Thank you so much. Good afternoon, everyone. First of all, I would like to really thank the organizer for this great event. I think it's very important to have this exchange between the academics and the private partners. Okay, so today I'm going to really present to you why messenger RNA is really can be considered as a game potential for the futures of the biomedicine. Is it working? Okay. But before I start, please allow me to really give a kind of short introductions of this new structure that the INSERM asked me to lead on mRNA. So it's called Accélérateur de recherche technologique, ARN messager, which is mRNA. And this is actually the fourth that INSERM built in France. And the goal is really to have an accelerator research unit to support innovation and translation of mRNA-based therapeutics and vaccine. So the mission we have is to accelerate the development of mRNA technologies. When I mean this, I mean mRNA structures, but also formulation. And for that, they gave me a capacity of innovation, meaning skills, HR, but also state-of-the-art equipment. And to develop also innovative project on mRNA, based on mRNA, therapeutic and vaccine. And here, it's of course a collaboration between academics or private labs. And last, complement or synergize, of course, the skills and what all of the partners have to really foster the development of the strategy. So the IRT-IRNM is really here to make a de-risking experiment based on using a clinical relevant studies. Then we have an interaction, we exchange know-how and procedures with our partners. And of course, then we have a high support, big support of the INSERM transfer, and which allows us to valorize via the valorization office of the partners. So today in the lab, we have a lipid and polymer-based formulations. I'm just going to show you very briefly here. For instance, it's a mRNA anti-cancer vaccine. It's done in a mice by IV injection. As you can see here, these are the mRNA coding for the antigens, and this is a control. You can really see that it's really working in terms of growth of a tumor, but the same for the innate immune system. And another one is, for instance, a delivery of mRNA in AK cells. So here, it's a protein replacement. Instead of using interleukin-2 protein, recombinant protein, we are transfecting the cells to support the proliferation. And as you can see here, it is really, you have exactly the same as the cytokine addition. And the last is a experiment on gene editing, where you edit with Cas9 mRNA and guide the expression of a specific protein. And you can see, depending on the type of formulation you are using, you can have a very, very good silencing of the gene. What is really nice with our formulation is the fact that they are very non-toxic, and you can make overnight incubation. And this is for cell therapy. You don't have, you can do it during the evening, go back home in the morning, take the cells. So welcome to discuss. You are very welcome to discuss if you would like to. Okay, so just to introduce you, my task today is just to introduce a little bit the session. So you know that the RNA-based medicine, there are different types of variety of strategies. I am going, as I said, focused on mRNA, but also on gene editing strategy. Philippe Barthélemy will actually talk on therapeutic oligonucleotide. And Nathalie Millier will introduce you the lipid-based nanosystem, because we need delivery with all of those different types of molecules. And as an example, I would like to show you clinical trials. I selected clinical trials that are going on just to show you that, yes, it's really important. And yes, I think we can do something which is really, really important for the health. Okay, so all of us agreed that since the success of this COVID-19 vaccine, but also there are a lot of preclinical studies, which is really a proof of concept in a different application. This is working very well in a different field. And also clinical trials, very, very promising. We really agreed that the mRNA holds a real promise as a new biologic. They are very interesting because you can actually deliver personalized strategy, therapeutic strategy, but also universal one. So you can really make this fine-tuning. And you can use it for immunotherapy, of course, but also gene editing, as I said, protein therapy, which is really very important in terms of metabolic disease, for instance, and also reprogramming. The benefit of the mRNA over other modality, most of you know it, it's a rapid and flexible production, as I've seen with the COVID-19 vaccine. The fact that you don't have to go to the nucleus is really very important because it's going to the nucleus stages. And of course, it has a safety issue because there is no gene from bacteria and very, very low risk of insertional mutagenesis. The productions of a protein with mRNA allows you to have all of the post-translational modification. It can be very, very difficult. And if you have antigens, you can have all of the epitopes. And of course, today, we can actually have also a prolonged expressions of the mRNA. The design of the mRNA, I'm not going to, to stay longer on this slide, but just, you have a 4-part, the cap, the non-transduced regions, coding sequence, and the poly-A. All of those structures, we know how to put them. We know how to add them. But still, I just want to say that the structure or the sequence on the 3' and 5' UTR are actually still, we know what you have to put for the vaccine, but if you would to use it, for instance, for protein therapy, I think there is much studies to be done, redone. The interaction between the cap and the poly-A, we also have to work a lot on this to really understand, do we need this? Is it possible? Which type of applications? The cap, for sure, we have to use a cap1 because cap1 is the one that we have on our mRNA. But I just want to say that this is really very dependent on the application you are using, on the type of cap1, cap0 you are using. So the mRNA structure that we are producing is a bit different than are actually in a prokaryote and in lower eukaryote. It's cap0. And I think Katalin Karikó, you are aware, but Katalin Karikó and Jill Vassman, they have been awarded of a Nobel Prize because of their milestone work on the modifications of the nucleoside, which is really very important. Karikó found that if she incubated different types of synthesized mRNA without modification, there is an increase of TNF-alpha secretion, leading to a blockade of the translation. So she had this idea to look at what are actually in the mRNA or RNA that we have in our cells. And she found out that actually the base are modified, part of the epitranscriptomic modification. And when she added different types of modification, she found out that in some types of modified nucleoside, there is a shutdown of the inflammation. And as a consequence, there is an increase of the expression of the mRNA. In this case, it's a luciferase. But you can also see that this modification is very dependent. I mean, the increase or the effect is dependent on the type of modification. We can talk on that later. So we have an mRNA which has a immune stimulation feature very good for vaccination. But again, you need to have a balance, but you have you can actually, dampen this by modification. But again, we need to identify and to select which one is the best. And that's why there are a lot of work to have to be done. A good balance between this mRNA and this delivery system, people forget about this, but the delivery system does really, the nature does matter. So today we can really, from this you can have a very high expression, long duration. So how do you produce it? Today, the only production we have is in vitro synthesis. And here you see, I'm not going, all of this, very different steps, from the plasmid production to the mRNA, the in vitro transcription. The challenge here is the complex supply chain and also the purification, because an mRNA which is not very well purified is really very difficult to express, even though you are doing modification. So different colleagues are working on cutting strategies to do this. The first one is the one that was made by my colleagues from Belgium, Karin Brechtpot and his collaborator. You can actually use a short mRNA that you produce by PCR if you would like to express a short epitope, a small epitope. It avoids you to use bacteria. You can also, instead of having, very difficult, you can also make an automated production using a factory-in-a-box approach. And here, it allows you to make in one step, you can have synthesis, purification, and this is actually an example from Quantum Bioscience, which is their intensified machine. The last one is a bioproduction in a cell factory. I'm just going to show you what we've done in my lab. It was a patent we have. There we decided to use a yeast to reproduce the mRNA. Of course, it's a little bit challenging, but I'm very glad because we have a European project on that, and we start to have a very nice data. And in France, we have bioRNA who actually took this patent, and they are actually building this as a startup, exploiting this as a startup. I'm very glad to see that there is a discussion between my partner and them to, speeding up the de-risking the technology. The application is by in vitro or in administration or in vivo. I'm not going to talk long about that, but just want to show you in my last minute I have. First example, mRNA cancer vaccine. One formulation using just a very simple liposome, using an messenger RNA, which is a Cap0, but a stabilized one chemically, and four mRNA coding for 4 melanoma antigens shared by different patients. As you can see here, it's a melanoma IV injection. It is one of the data. There is, of course, a responder. There is a non-responder. And here, it has been shown that you can actually use an anti-PD-1 as an adjuvant to enhance the efficiency of this vaccine. It's now in a phase 2 by BioNTech. And it's done also for pancreatic cancer and different types of cancer. Second one, exactly against melanoma, metastatic melanoma. In this case, they are using LNP plus Cap1 and modified mRNA by intramuscular injection. And you can see here, it's a 34 neoantigens. It allows me to tell you that you can encapsulate a lot of molecules in LNP. Every 3 weeks, you see this data. Very, very nice. And today, this is in a phase 3 49% of reductions, and it's really very good. Again, it's also exploited for lung cancers. Another one is just to show you that you can also use it for CAR-T therapy. And here is, I think it has been discussed this morning. It's Cartesian Therapeutics is doing this. So it's a mRNA engineering B cells via targeting BCMA. And you can see here the very good benefit of this is that you don't have any, CRS syndrome. See, there is no dose limit, and it's really very interesting. Concerning the gene editing approach, the colleagues this morning talked a lot about this, so I don't want to show you what we have to do when you do this with mRNA. Clearly, we need to really have a production of long modified nucleic acid for do a prime editing, because I think prime editing is this editing used in reverse transcriptase. Again, you have to optimize the lipid and polymer for fat, otherwise it's not working. What is actually in the progress today? Casgevy, I'm not going to talk it because it has been mentioned. 2, Intelia therapeutic, if the transthyretin, you see, just one dose instead of doing it periodically, just one dose. You have a very the expression is really downregulated for a long time. And there is another one, is a calcrein B1 for angioedema, and it's also very, very promising. They are in phase 1 and 2. A prime editor is for phase 1. Here, in all of this, the Cas9 is given by mRNA. So I think we can really see, I hope, that we have now real opportunities to exploit this molecule. But of course, we have challenges. The opportunity is the versatility of the technology. As I said, you can really make many, as 30, 39, 20 mRNA. You can encapsulate guide, you can encapsulate another types of molecule. The durations of mRNA has been addressed already. So you can use self-amplifying mRNA, allowing you to, then you add the replicase that can amplify the mRNA. Of course, the mRNA becomes very long, starting from 9 kb. So you have to find out the right delivery system, and sometimes it can be, there are some immunity. The Tron decided to do it, cut it in 2, trans-amplifying. One is for the replicase, and one is for the gene, the mRNA that you want to express. And it's working very well because you are reducing the size. And you can actually spare the dose for your treatment. Circular RNA, of course, it's very interesting because you have a loop, high stability. At the beginning, we thought that you don't need to have any modification, but the last paper showing that indeed, if you want to have a high increased expression, you need to have some modification. The challenge for me is really, as I already said about the epitranscriptomic modification, I'm not going back over that. The targeting is a real challenge today because most of the LNP goes in the liver, you have to dedark it from the liver. Hopefully with these chemical vectors, we do have different types of conjugations that you can apply for. Then you have to think about how can we actually, what type of ligands do we have to address? Do we have something that can really not induce in any immunity? And for the mRNA, you can also work on the mRNA to allow the mRNA to be degraded when it's not in the right place. It's using a microRNA target. So from the classic manufacturing, all of us agree that we need to use an AA enabling manufacturing. We are doing it in a private company. We are start doing it also in a lab. Predictive and efficient model to reduce the time of risk. And of course, the last one we have to do as an adaptive of manufacturing if you would like to work close to the bed of the patient. For that, I would like to thank you for your attention and thank you to my support. Thank you so much.
Thank you very much, and we'll find you back, of course, during the round table.